Today I am trying to discuss with one of the important sensory modality that provides us the sensation of position, the body position or the limb positions in space. This sensation has been hijacked by the other posterior column sensations. In the other way, it has got a less attention in terms of its receptors, the pathways, the destinations, as has been described for other posterior column sensations like touch, pressure, vibration, stereognosis, tactile localization, tactile discrimination. I scanned several textbooks. I could not find, barring one or two, I could not find a very clear presentation of this sensation. Hence, I have made an effort to provide you the comprehensive aspect on proprioception. Think about doing any action or movement, just as in case of a bicycle riding. We do it effortlessly. We do it while talking to our friends, laughing at the jokes, seeing the scenery around us, or listening to the music, while performing, while moving, or while pedaling the bicycle. Such a effortless, such unconscious things are possible because of the proprioception. Proprioception, by definition, includes the sense of position and movement of our limbs, senses of muscle force and effort, and sense of balance. So there are three components which have been provided here. In nutshell, it is the sensations of the body position or the limbs position, limbs or extremity position in space in reference to head or body. Regarding proprioception, von Helmholtz proposed that it is the sensation begins from the cerebral cortex. Later on, Sir Charles Sherrington proposed that it is the sensation starting from the spine, the spinal cord or the body. Now, both of these parts are true. It is now placed as a sixth sense in the sensory modality. This is also known as a kinesthesia. The proprioception allows us to carry daily routine tasks successfully, unconsciously, effortlessly, with ease. In the absence of feedback from the proprioceptors, think how awkward our actions would be. So we will not be able to perform the actions as we are doing it. What are the Types of proprioception. The proprioception involves both conscious and unconscious efforts. The conscious proprioception, just like if I ask you to touch your finger, you will touch. That means identifying a position of the joint or a part, or the part moved when asked. This is possible when we are conscious. That means the information is going up to the sensory cortex and there it is processed. There are two components in the proprioception. One, the stationary, and the dynamic, that is during motion. That means when we are asking, when I ask perform certain actions, where is that body part, I can still recall. Finger nose test, finger test with closed eye is one of the example of the conscious proprioception. Suppose if you have been given certain objects in your hand and asked you to tell the weight of the object, what do? You just toss it up on either side, on this hand and that hand, and finally you come up with a particular approximate weight. So that means tossing provides that proprioceptive activity, be proprioceptors activated, and then that provides the weight. We will also know the severity of the muscle action when in motion. Many other functions, but that requires the conscious state. That means the sensory cortex should be functional. The unconscious proprioception involves number of things happening while we are not conscious. Just for example, even when in motion we are thinking about and we are looking at certain things, suddenly somebody pushes, you try to bring back your position. In the unconscious proprioception, the position and movement of our body and its part, including dynamics. Dynamics includes the mass, the forces acting, the gravitational pull, and in relation to timing of the particular sequence of action. All that information is vested in the unconscious proprioception. This unconscious proprioception is processed at cerebellar structures. In comparison to the conscious proprioception, they are processed in the parietal cortex or sensory cortex, further with the somatosensory area, association area. The unconscious proprioception necessary for segmental, suprasegmental, vestibular and cerebellar reflexes or reflex alterations for posture and equilibrium. Thus, these unconscious sensations are integral of number of postural reflexes including the writing reflexes. Similar to the conscious proprioception, there are static and dynamic components of the proprioception that I will be dealing in the subsequent slides. What are the receptors involved in proprioception? I have listed here in this table and for the conscious proprioception you can have those joint capsule refining end organs, joint capsule Pacinian corpuscles, stretch sensitive nerve endings in the joint capsule. Whereas in the unconscious proprioception, all the 6 items listed here will be participating in one or the other way. Just I am briefing the muscle spindle. The muscle spindle or the intrafusal fiber, this is located within the muscle parallel to the extrafusal fiber. These are encapsulated receptors. You have nuclear bag fibers, nuclear chain fibers, you have a static intrafusal fibers and dynamic intrafusal fibers. What exactly they do is they know the muscle length, they monitor the muscle length. That means a change in the length would be detected. Also, the rate of change, that is the speed of contraction, is also detected by these muscle spindle primary endings. The function is to maintain the length of the muscle and regulate the speed of contraction. These receptors are carried by group 1A fibers or A alpha fibers. These are fast conducting fibers with a velocity around 100 meters per second, and these are slowly adapting receptors. In the intrafusal fibers, we have on the polar end the secondary endings. These are annulospiral endings or flower spray endings. They also detect stretch, and their function is to reduce the muscle stretch or modulate the muscle stretch. They are carried by the group 2 fibers with a conduction velocity somewhere around 50 to 60 meters per second, carried also known as A beta fibers. These are also slowly adapting. We have another proprioceptor located in the tendon organs and also in the muscles. These are also known as a Golgi tendon organs or Golgi organs. These are encapsulated receptors with a collagen covered, and they are also having a spray endings. That means they are present in different forms. They sense the excessive stretch of the muscle or excessive contraction of the muscle. Actually, they detect when the muscle contract very rigorously, very severely. So that means they try to regulate muscle tension, muscle activity and twisting so as the muscle is not ruptured during severe contraction. These receptors are carried by group 1B or A alpha fibers. These are also fast conducting fibers with 100 meter per second conduction velocity. Just to brief you about the terminology 1A, 1B in sensory modality, the A and B suffix indicates the threshold of the fibers. Group 1A fibers have a low threshold. Group 1B fibers require a higher threshold for stimulation. Accordingly, they are called group 1A and 1B. These are also slowly adapting receptors. We have described the Ruffini end organs. They are present in the joint capsule all around. They are encapsulated. As has been described earlier, they carry touch, stretch, traction, and pressure. They detect the joint angle and torque. That means they regulate or modulate the joint angle and the torque change in the angle. That means the twisting of the joint. They are carried by the group 2 fibers or A beta fibers with a conduction velocity around 50 to 60 meters per second. They are also slowly adapting receptors. We have another receptor in the joint capsule, that is a joint capsule Pacinian corpuscles. These are encapsulated, layered receptors. They detect the joint movement. They detect the direction and velocity, or they modulate the direction and velocity of the motion carried by group 2 fibers or A beta fibers. They are rapidly adapting receptors, as already been discussed. There are other group of nerve endings. These are free nerve endings located around the joint. These are free nerve endings. They detect touch, pressure, and they modulate excessive stretch and force on the joint, and they are carried by small diameter fibers, small diameter myelinated fibers, A delta fibers, or group 3 fibers. These are also slowly adapting. These are the receptors for the proprioception, both conscious or unconscious. Here I am showing you on one side the muscle spindle. I was trying to describe you the central and part. We have nuclear bag fibers, nuclear chain fibers. That means that stick-like things, nuclear chain, and the bulb-like things are nuclear bag fibers. And in the polar region, we have those group 2 fibers or secondary nerve endings. You can see that. So we have in the bottom here on the left side, Golgi tendon organ. This is just like a ramifications of these terminals to the different components of the Golgi tendon or fascia, and they detect the stretch or traction. Then on the right side we have the joint here. In the joint we have the Golgi tendon organs also, because fascia is there, and we have Ruffini end organs, the Pacinian corpuscles, and the free nerve endings, so on. Just to remind you about the muscle spindle, at rest it is firing not as much. In the bottom, there is a stretch. When there is a stretch, the primary endings will provide continuous burst of activity. The spurt of activity you see on the top location. Then secondary endings, they increase the discharge continuously or tonically. The secondary endings, in fact, they are not adapting as much as of the primary endings. In the bottom, we have these Golgi tendon organs, the receptors. You can just see when the muscle contracts, when it's the contraction of the muscle or a tension developed by the muscle at its peak, you will see the discharge. As the muscle tension is decreasing, the discharge decreases. So that means it prevents the rupture of the muscle fibers if it is rigorously contracting. What are the pathways for conscious proprioception? Already we know, because these are conscious proprioception are carried by the posterior column sensation. Just briefing here, the receptors are Ruffini end organs, Pacinian corpuscles, free nerve endings around the joint, carried by group 2 or A beta fibers through the dorsal root ganglia to the posterior column. Then, after giving a collateral in the spinal cord, they ascend in the fasciculus gracilis for the lower limb, fasciculus cuneatus for the upper extremity as described earlier, and relay in nucleus gracilis and nucleus cuneatus in the medulla, respectively. After the relay, the second order neurons generate, or these second order neurons, the axons of these second order neurons, they cross to the opposite side and ascend as a medial lemniscal fibers to relay in the ventero-posterior-lateral nucleus of the thalamus. Third order neuron starts from thalamus, and thalamocortical fibers are relayed in sensory cortex area in the parietal cortex, area 312, or what is called the somatosensory area number 1, with a definite topographical representation as in homunculus. The outputs of S1 are relayed to S2 and somatosensory area 5 and 7 for further analysis, wherein we can tell about the joint positions or the position of the limb in a particular type of motion, when a particular time of motion are either static or dynamic, or we can also tell about how severe is the force of contraction or how severe is the activity. That also, all these parameters are all attributes regarding this motion or this thing, how much is twisted, how much bent the particular joint has taken place. All those attributes, you can see them here in this analysis. For this, you see the details in the posterior column sensations in my previous lecture. Just to summarize, here I have taken the picture from the previous lecture. The receptors for touch, pressure, proprioception and vibration, they are coming here and they ascend in the fasciculus gracilis and fasciculus cuneatus, you can see there, and reach without crossing up to the medulla in the dorsal column fibers, the fasciculus gracilis and fasciculus cuneatus. In the medulla, they relay in the nucleus gracilis and nucleus cuneatus, then cross over to the opposite side in the medulla and ascend to thalamus as a medial lemniscal bundle. In the thalamus, they are relayed in the ventroposterolateral nucleus of the thalamus. From there, the third order neurons begin and they reach the parietal cortex in a definite topographical location. Subsequently analyzed with the S1 and S2. The details you can go with the posterior column, any other posterior column sensations, where the attributes of a particular motion, action, joint position are all monitored or can be detected or specified. Now consider the unconscious proprioceptive pathways. Receptors are already described. They are from muscle spindle, Golgi tendon organs, Ruffini end organs, Pacinian corpuscles, and free nerve endings, and so on, around the joints, tendons, and the fascia. The fibers carrying these receptors include the large diameter A alpha fibers, group 1A, 1B, A beta fibers, A delta fibers that will reach through the dorsal root ganglion. Basically, two different type of pathways reaching the destination point, that is cerebellum. One is direct pathway, another is indirect pathway. The direct pathways include four pathways. These are ventral spinocerebellar tract, dorsal spinocerebellar tract, rostral spinocerebellar tract, cuneocerebellar tract. Ventral and dorsal spinocerebellar tract carry information from lower limb and trunk. Rostral and cuneocerebellar tracts carry information from upper limb and neck. The ventral spinocerebellar tract and the rostral spinocerebellar tract, they carry similar type of sensation. Dorsal spinocerebellar tract and cuneocerebellar tract, similar type of sensation. These are the direct pathways. The indirect pathways include the pathways reaching to inferior olivary nucleus, that is spino-olivary tract. From the spino-olivary tract, they will reach to cerebellum as a olivocerebellar pathways. On the right hand side, I had given the plan of various tracts in the spinal cord transverse section, and on the lower various laminar pattern of the spinal gray matter is provided. We will consider the direct pathways. One of them is the dorsal spinocerebellar tract. This originates from the proprioceptors of the lower limb and trunk. That means the spinal segments, the thoracic and the lumbar segments are involved. Through the posterior root ganglion, the fibers enter dorsal horn gray matter. They relay in 1, 5, 7 lamina of the spinal cord, especially on the marginal side. And this nucleus is known as a nucleus propriosis. The second order neurons from these, they will traverse in the lateral funiculus on the same side as dorsal spinocerebellar tract, because they are placed on the posterior side or a dorsal side. That is marked here, that is shown in the transverse section of the spinal cord also, that is shown in the left hand figure where the are going. The second order neurons ascend up to the medulla. In the medulla, via the inferior cerebellar peduncle, they relay in spinocerebellum as mossy fibers. The area of the spinocerebellum represents the lower extremity. The ventral spinocerebellar tract also receives inputs from lower limb proprioceptors, especially from segments, lumbar and sacral segments. Major proprioceptors for this tract are the Golgi tendon organs. These fibers through the dorsal root ganglia enter the dorsal root and relay in Clark's nucleus or Clark's column, marked as a blue area in the gray matter. After the relay, the second order neurons cross to the opposite side, ascend in the lateral funiculus anterior side, marked as a cross there in the transverse section of the spinal cord and marked as a fiber there with a cross. These fibers ascend in this tract, that is ventral spinocerebellar tract, reach the pons and relay in the spinocerebellum. Again, what happens? That enters into the superior cerebellar peduncle and crosses again to the ipsilateral side. That means the sensation originating from the left, now it crossed, ascend as in the right side, ascended up to pons. Again in pons, it crosses one time and reaches the same side, left side of the cerebellar hemisphere, and relayed into the spinocerebellum. The fibers are again the mossy the fibers these are carrying or entering the spinal cord as mossy fibers, representing the lower extremity. Rostral spinocerebellar tract, it is from the upper limb and the neck proprioceptors. They relay, as in case of posterior spinocerebellar tract, in lamina 1, 5 and 7 of the spinal cord, ascend in the same side without crossing. However, these fibers are located rostrally in the dorsal spinocerebellar tract and they reach up to the medulla. After reaching the medulla, through the inferior cerebellar peduncle, they relay in the respective nucleus of the spinocerebellum, representing the upper part, that means the upper limb and the neck. Cuneo-cerebellar tract, these carry the proprioceptive inputs from the upper extremity, especially the upper limbs and the neck. They ascend with the fasciculus cuneatus on the same side, along with the other posterior column sensations, up to the medulla. From the medulla, they will relay, these fibers relay into the lateral cuneate nucleus. The lateral cuneate nucleus is also known as the external cuneate nucleus or the accessory cuneate nucleus. And the axons of these second order neurons enter the cerebellum via inferior cerebellar peduncle and reach the spinocerebellum as mossy fibers, representing the upper extremity of the same side. Coming to the indirect path, carrying the unconscious proprioception, these are Spino-Olivary tract. The origin of the Spino-Olivary tract begins with the proprioceptors from all segments of the body. That means starting from the cervical to the lumbar sacral regions, all the muscles, muscle spindles, the Golgi tendon organs, the ligament, free nerve endings, so on, all the receptors are coming with this tract. They reach through the push root by the dorsal root ganglion, and then in the gray matter they relay in the lamina 3-4 of the spinal cord on the lateral side. From here, the second order neurons cross to the opposite side, ascend as a Spino-Olivary tract, indicated by the cross. In the medulla, these fibers relay in the inferior olivary nucleus. The inferior olivary nucleus receives a variety of inputs from the different areas, from reticular system, vestibular system, and from the cerebral, pontine, and the collicular. All inputs are given here, and from here the olivocerebellar tract begins. These olivocerebellar tracts, these fibers, pass through the inferior cerebellar peduncle and relay in the spinocerebellum as climbing fibers. If you are looking at, again, there is a 2 times crossing from once at the spinal cord level, the Spino-Olivary tract, from the Olivary tract, the second order neurons cross to the opposite side, and again from the olivocerebellar it crosses to the opposite side. That means whatever the information the cerebellum receives, it is from the same side of the body. All the 4 direct pathways ascend or reach the cerebellum as a mossy fiber, whereas the olivocerebellar fibers as a climbing fiber. We will look into it in the next slides. In this table, the details of all the pathways carrying the unconscious proprioception are summarized. I can quickly go through and highlight those specialties. Ventral spinocerebellar tract, originates from L1 to S5 segments. Parts from lower limb and trunks only, it receives the proprioceptive inputs predominantly from Golgi organs. Group 1B fibers. They rely in dorsal root gray matter 157 lamina and then cross to the opposite side, ascend as ventral spinocerebellar tract and then reach the pons through the superior cerebellar peduncle. There is one more crossing, second time it crosses and reaches the cerebellum, representing the lower extremity. The fibers reaching the cerebellum as mossy fibers. In case of posterior spinocerebellar tract, C8 to L3 segments are involved. All the proprioceptive inputs covered by these segments. Trunk and lower limb are the part supplied, predominantly muscle spindle, group 1A fibers. Relayed in Clark's column, they do not cross and ascend ipsilaterally as posterior spinocerebellar tract, reach the medulla through the inferior cerebellar peduncle, reach the spinocerebellum as mossy fibers. In case of rostral spinocerebellar tract, the segments involved are C4 to C7, mainly upper limb and neck. They receive proprioceptive inputs predominantly from Golgi organs, 1B fibers. The nerve enters through the dorsal root ganglia and then in the dorsal horn, lamina 157 of the spinal gray. These fibers do not cross and they ascend ipsilaterally on the rostral side of the posterior spinocerebellar tract. That means they are placed rostrally. They reach the medulla through the inferior peduncle. They go to the spinocerebellum, representing the neck and upper extremity region, and in the cerebellum they are as mossy fibers. The cuneo-cerebellar tract, they originate from C2 to T7, neck and upper limb proprioceptors, predominantly muscle spindle, group 1A fibers. They travel along with the fasciculus cuneatus in the posterior column and relay in the lateral cuneate nucleus in the medulla, and the second order neurons from the lateral cuneate nucleus, the axons, they reach the ipsilateral cerebellum through inferior cerebellar peduncle. These fibers in the spinal cerebellum as mossy fibers. Spino-olivary tract. This receives inputs from all segments of the spinal cord from the entire body. Proprioceptors. They reach the dorsal gray matter through the dorsal root ganglion and relay there in lamina 3-4 on the marginal side. Here the second order neurons cross to the opposite side and ascend as inferior olivary nucleus, inferior olivary tract. From the olivary nucleus, the fresh tract originates, that is olivocerebellar tract, and via the inferior cerebellar peduncle, they will reach the spinocerebellum as climbing fibers. Now, important question is, these spinocerebellar fibers and olivary fibers, where they end? In the cerebellum. Let us look into the cerebellum in a physiological perspectives. It can be divided into three major parts. Outer part of the cerebellar hemisphere, known as a lateral cerebellum, this is connected with the cerebrum in terms of the motor planning and coordination. That is cerebrocerebellum. Then central part, as indicated by that star, spinocerebellum, that is vermis and paravermis area, that is spinocerebellum. This spinocerebellum receives inputs from the axial muscles as well as distal muscles which are doing action. Third part of the cerebellum is connected with the vestibular system, that is vestibulocerebellum. That forms the flocculonodular lobe. Now the cerebellum, each part of the cerebellum has a nucleus, the deep cerebral, what is called a deep cerebellar nuclei. The cerebral part, that is a lateral cerebellum, is having dentate nucleus. In case of the spinocerebellum, there are two nuclei, one nucleus interpositus and the vermis, the central portion which is receiving the axial inputs, that is called vestigial nucleus. The flocculonodular lobe, that is have a vestibular nucleus. These are the nuclei on the other side where they will give. If you are coming back, vermis is connected with the axial muscles, all the proprioceptive inputs from them, and then the output also reach to them. The output of the vestigial nucleus will go to vestibular nucleus and reticular nucleus, which try to govern the posture and gravity through these axial muscles. Whereas the distal muscles, where these are muscles for fine movements, running, walking, talking, and even writing, all these distal muscles are performed by the distal muscles. Nucleus interpositus is involved. The output from the nucleus interpositus goes to red nucleus, collicular nucleus, and reticular nucleus. They modulate the distal muscles. They try to dampen or prevent the overshoot or undershoot of the action, and also they govern the eye movements. Direct pathways enter the cerebellum as mossy fibers, and the olivary fibers, that is the indirect pathway, they enter the cerebellum as climbing fibers. We will see this thing in the next slide. This is showing the representation of the various proprioceptors in the cerebellum. You can just see there is a double representation. One, the head side down in the anterior cerebellum, you can just see that, and again this is a part of the in the bottom also there is a part of the anterior cerebellum that is a curved one. So now you have one side, it is upside down. The central one, the axial muscles are represented in the vermis. The limb muscles are represented in the little intermediate, that is the spinocerebellum, intermediate part of the spinocerebellum. Then you can just see the extraocular muscles and the head muscles are also there. It is in a reverse direction. That is how it is being topographically monitored. On the other side, there is a location in the center. The vermis have both sides put together. Then comes the limbs, one side, the other side, upper limb and lower limb, and the neck muscles. This is the typical arrangement of the distribution of the proprioceptors in the cerebellum. Similarly, the same type of motor activity is given by these group of fibers or two group of these group of nuclei. The proprioceptors are represented on both sides. You can just see on both sides there is a lot of connections, and but the fibers they reach up to ipsilaterally, then they get mixed up. Now we discuss about the processing of the proprioceptive sensation in the cerebellum. On the left hand side we have the cerebellar cellular architecture. The Purkinje cells, represented as PC, are the large neurons. They are the main neurons of the cerebellum. They give inhibitory output to the deep cerebellar nucleus, that is NC. The output from the deep cerebellar nucleus to the various other places is always excitatory. Thus, Purkinje cells modulate the output, whether it is going in bursts or it is diminished. The output of the deep cerebellar nucleus is excitatory to the respective area. The vestigial nucleus gives output to the vestibular nucleus and the reticular nucleus, because this is concerned with the posture, equilibrium and maintenance of gravity. The interpositus nucleus gives output to the red nucleus primarily, that is for the movement or the locomotion. Also the collicular nucleus, wherein the eye movement is monitored. Then reticular nucleus for coordinating. We have other cellular type, the GR, granular cell, that is another important cell in the cerebellar outline. Granular cells give a T-like projections, and these axons, they interdigitate or connect with the Purkinje cell dendrites. Purkinje cells have a huge dendritic arborization like a tree. These parallel fibers cut these branches, various branches of the tree, and at each cutting they will make a synapse, that is shown there with the one synapse. The granular cell output is excitatory, and this excitatory output to the Purkinje cell excites the Purkinje cell. The mossy fibers originating from the spinocerebellar tracts, the dorsal spinocerebellar, ventral spinocerebellar, rostral spinocerebellar, and the cuneo-cerebellar tracts, they ascend as a mossy output. The mossy output comes here, excite the deep cerebellar nuclei, and also excite the granular cell. Once it excites the deep cerebellar nucleus, there is a burst of activity going to the excitatory activity going to the respective areas as mentioned, either with a vestigial or interpositus nucleus outputs. Then granular cell, the input to the granular cell, the excitatory input to the granular cell provide the excitatory input to the Purkinje cell. And these Purkinje cells, once they are excited, they will restrict or they will send inhibitory inputs to the deep granular. So that means the action is began, then it is modulated, because there should not be too much of action. That means action has to be modulated. This is the modulation by the Purkinje cells. We have the basket cells. modulating the Purkinje cells. There are stellate cells which are not shown there, and we have the Golgi cells, GC, which will modulate the granular cell output. On the other hand, we have the climbing fibers. You see the climbing fibers there. They come and climb the Purkinje cells, and they will excite the Purkinje cells so that they will excite the deep nucleus and the Purkinje cells. First, the action begins, then climbing fibers abruptly stops. Overall, when action begins, we see that the first one to come is the MOSI input, because you have only one synapse there. Then second synapse is coming here, that is relaying its spinocerebellar tract, relaying into the deep cerebellar nucleus and the granular cells. So it arrives first, it begins the action, and once it begins the action, that action has to be stopped. Now at the same time now the climbing fibers come, because the climbing fibers have one more synapse there in the inferior olivary nucleus. From there it traverses into the inferior peduncle and to the cerebellum as climbing fibers, and it will come and excite this thing. So that means it reinforces the action initiated by the MOSI fiber, and then it will it will stimulate the Purkinje cell very strongly, and it will. So the overall action of the MOSI fiber and the climbing fiber is to smoothen the action. Thus it dampens or prevents the overshoot of the action. It makes it smooth. It requires, when we take up the cerebellum, we will go little more detail. This is how the cerebellar processing taking place through the MOSI fiber and the climbing fiber input, and both of them modulate the deep cerebellar nuclear output to the respective areas. What are the various clinical conditions associated with this sensation? I mentioned in the posterior column sensations, one condition is subacute combined degeneration, wherein there is a deficiency of the vitamin B12 and folic acid. Vitamin B12 is necessary for erythropoiesis, as we already know. In addition to that, it is necessary for the synthesis of myelin. In the deficiency of vitamin B12, the myelin synthesis is altered, and hence the large diameter fibers are affected. That is subacute combined degeneration. When large diameter fibers are affected, all the sensations carried by the posterior column are altered. So that means touch, pressure, vibration, they will be diminished or lost. Proprioception is also lost. Or any demyelinating disorders, may be viral, toxin, genetic. Just like multiple sclerosis. The Tabes dorsalis is another condition. The tertiary syphilis affects the myelin synthesis, and that would also affect the posterior column neurons. So that means all these sensations are altered. That means posterior column is affected. Other genetic disorders like Friedreich's ataxia. The one thing common with all these conditions is they affect the large diameter fibers. Hence the posterior column sensations are altered, while the pain and thermal sensations, which are not transmitted with the posterior column, are preserved. We will try to look into the various tests performed to evaluate the functionality of the proprioception. One of the tests is Romberg sign. To evaluate Romberg sign, the person is asked to stand with closed eyes and feet held together. Normally, healthy person is able to stand without any swaying on either side or falling. The persons with the posterior column lesions, they will sway on either side and even fall. So when you are performing this test, you have to be cautious to see that the person should not fall down and get injured. The inability to balance is due to the absence of proprioceptive inputs from the lower limb to the cerebellum. And this type of ataxia is known as a sensory ataxia. And when such a response takes place, this is called Romberg sign positive. Also, it is called Rombergism. On the other hand, there is another term called motor ataxia. It is due to the defect in the cerebellum. In this condition, person is not able to stand even with open eyes. So that means there is a defect in the cerebellum. Number two, we have to evaluate various spinal reflexes, because these spinal reflexes are carried by the afferents either originating from the intrafusal fibers or from the Golgi tendon organs. There are also known as a flexor reflex afferents. So all these reflexes, the stretch reflex elicited by intrafusal fiber, inverse stretch reflex by the Golgi tendon organs, or a crossed extensor reflex, or reciprocal inhibition, so on, are lost or diminished in posterior column lesions. Another important test to evaluate the function is to observe the gait. Person is asked to walk in a straight line. While he is walking, we observe the persons with posterior column lesions, they walk very clumsily, they look down and place the foot on the ground with a great force. Practically they pound or they stamp. That is why it is known as a stamping gait. Sometimes these persons lose balance and fall frequently. In no circumstances the person with the posterior column lesions will not be able to walk in a straight line with a closed eye, because he will sway on either side and even he will fall down. This is all because the absence of position sense, which is carried from the lower limb, is missing. I have referred following books as the base. However, most of the information has been researched and taken from number of sites which I have mentioned then and there. It is just in order to give a comprehensive view of the proprioceptive sensation. Hope it will be fine. In my next lecture, I will continue with the anterolateral column sensations pathway processing. Thank you. Here I have given some assignments for your own assessment. Just go through them and complete them and try to evaluate yourself. And these are some of the assignments which you can keep working. Maybe there will be little longer questions. You can just look at that and try to study them backwards. Thank you very much.